yeah, I'm really very pleased to see so many people replying to our invitation, and I'm happy to extend, in the name of ELDICO, our very warm welcome to all of you. I think for some of you, or at least for me, it's one of the very first in-person meetings again, which is indeed a great pleasure. I notice you never know if you meet like this or like that. Yeah, but we will get used to regular handshaking again, I suppose. Definitely, sure, we will manage. Yeah, we set an interesting program together for you. You all got the information. I hope you like our handout with some additional information. But otherwise, I would like to start and introduce Professor Hans-Florian Zeilhofer as a first speaker to you. Hans-Florian is at the University of Basel, head of the Innovation Office. From other tasks, he retired last year, but still very active and committed to the university, the academic life, and in particular the Innovation Office, and in that sense also Swiss Innovation, which is kind of our host here in this building, not only today, but also for the application lab that we have downstairs. All of that is in collaboration with the Swiss Innovation Park in Basel here, Basel area. Hans-Florian, very active in translation of scientific results into entrepreneurship, and that will also be his talk today. I don't see this title here. The transformative potential of innovation platforms. Thank you, Hans.
Thanks.
You need? No.
Ladies and gentlemen, dear friends, dear ELDICO team, again, welcome. Welcome here in this place, which is really a great place with a great history as well. I started here in 2015 with the University of Basel, and we moved the Department of Biomedical Engineering here in this place. And we moved it because it should be a pilot, a pioneer, the first in this place for enabling the new Switzerland Innovation Park, which was accepted to be in this area, in the neighborhood here. And we wanted as early as possible come out and feel the spirit and bring a spirit into this place. And in this building, we had a lot of startups, research, in an ecosystem, I could say, with big companies as well, and companies in the neighborhood, like today Idorsia, at that time Actelion, which started here in this building, Mr. Klosel told me. And I remember also very well when about 2 years ago, the ELDICO team came first time here. And then in order to show us what is your new technology in imaging, we had here a small symposium, but highest class, I would say. And this convinced us that we need your technology, we need you as a startup here in this ecosystem hotspot of life science. And then we had negotiations, and you asked me, and at that time I didn't take the hat of the university and not the hat of Innovation Park, Switzerland Innovation Park. I said, as a young entrepreneur, because I made several startups as well, you need to go and to come here. Here is a hotspot, the best place in Europe. But at that time, I said, it will be the best place, the first place, the number one in Europe. And you have to be with your technology in this number one, cutting edge has come to a cutting edge place. And at that time, it was a green grassland. And I showed you and inspired you for the image what will be here in a couple of years. And today, if you go over there, you see already the main campus ready. And the first company will be Basilea. Basilea with a history as well, with 4 Nobel laureates. They will move into the new building now in June, and our university will follow in November. And I hope you also will follow very soon, because we need you in this ecosystem. Then it will be an ecosystem. An ecosystem is also a sort of family. And I talked to you at that time, first time, of a platform. A platform, and this is from my side, from the university, also a task or a challenge for you. I would like you to be similar as we were with our department, a pioneer, a nucleus for this Switzerland Innovation Park. You should be the nucleus of an imaging, high-class imaging platform in this innovation area. Life science needs this. You're connecting, you're a glue then. And a platform is something where everybody who likes can dock on. And that means, and we saw this in the past, a company like Idorsia, Novartis, Roche, J&J, Merck, they all can come here on the platform. There is no more competition at the moment where they meet here in this neutral zone. And I love it because they have an inspiring atmosphere together with researchers. You have top-class research around you. You have different universities, because we are not only University of Basel, our university has a university network as well we bring here. And we have the industrial, the entrepreneurial network. We have the politics here, the Standortförderung, which really is very important from 3 cantons, not only Basel-Stadt, it's also Basel area, and it's a canton of Jura. And we have also a strong cooperation in the Upper Rhine area with Germany and with France. So this is the right time that you start here with the inauguration of your technology, of the first device here in Basel. Thank you, and I wish you good luck. And I promise you, you will be in a couple of years as successful as many others who started here.
Yeah, thank you very much, Hans Florian. Indeed, we're very much looking forward to settle up here, and in particular moving to the new building, which will happen around the summer. I heard that even in July we might be moving in already, which will give us a little bit more space and a little bit nicer even application lab. Although we are already very happy having the lab downstairs and being part of this ecosystem, and in fact with all the companies that you named, we have good contacts already. But being really settled here will only strengthen these collaborations with these companies. So in that sense, we are very much looking forward in settling here in the campus. And thank you for the opportunity and the possibility, having the logistics and the building and all the support from the Basel area to enable us as a startup to realize our dream in the end. Yeah. Okay, thank you for that. We move on to our next speaker, and I'm glad to introduce Dr. Christoph Boss as a next speaker. Christoph is head of drug discovery chemistry within Idorsia. He's a member of the leadership team at Idorsia, and very much involved in synthetic organic chemistry still, and that's of course an important part of Idorsia, but there's also a topic that involves us very much. And he will talk about challenges for modern drug discovery and development as our keynote speaker. Welcome, Christoph, and the floor is yours. Yep.
Okay.
Next slide would be yours.
Thank you very much.
Yeah.
Okay. So maybe as we started with history, let me add something to the historic aspect of this building. So it feels a bit like coming home. I started in this building on this floor, 1st of February 1999, as one of the first lab heads at the Electron Pharmaceuticals. So now we are at Idorsia over there, but still it's like a little bit like coming home. Then maybe also a thought that I had when I got the invitation to talk here. I was kind of wondering why the heck did they invite me? Because I really, I'm blunt with respect to the technology that you are discussing today, with respect to all the machine aspects and behind, I have no clue. So maybe it was because when approximately 30 years ago I started my PhD at the University of Bern, I joined the educational course in Hans-Beat Bürki's group about X-ray crystallography, but I did my thesis in organic synthetic chemistry. So maybe somebody knew about that and thought, okay, he has some background knowledge, very, very old background knowledge from very, very old technology, so he could maybe make it here. But I think let's come to the more serious part. So what I would like to do today is I would like to give you a certain insight into Idorsia. Then I would like also to tell you why we think 3D is important for drug discovery and for drug discovery chemistry. And then I would also like to mention a few aspects where we think that we can potentially benefit from the investment that we already made into ELDICO, because so far I think we only transferred money and we didn't get much back. So hopefully, hopefully in the near future we will get back data for the investment that we are making. We are absolutely convinced that this is a good investment, otherwise we wouldn't make it. But now let's move forward together. So. One slide is missing. Okay, so Idorsia is actually built on 5 strategic priorities. And the first strategic priority is bringing 3 products to the market. Where are we with that? So we have daridorexant, which is a dual orexin receptor antagonist used in the treatment of insomnia, which was recently approved in the United States, and we will hopefully soon start selling it under the brand name of QUVIC in the US. Approval process is ongoing in Europe, in Europe. We have clazosentan, shown here, an endothelin receptor antagonist, which successfully terminated clinical phase 3 trials in Japan and is now approved in Japan as PIVLAZ. So we will hopefully also soon start selling this one. Then we have aprocitentan in collaboration with Johnson & Johnson for the treatment of resistant hypertension. It's a dual endothelin receptor antagonist. Phase 3 clinical trial recruiting is terminated, so we are waiting for the results to be finished in the analysis and then hopefully soon published, and we for sure hope that it's going to be successful. Then we have lucerastat, where we have finished phase 3, which resulted in not met primary endpoints, but a lot of positive aspects that we are negotiating with the authorities. Then we have other products like selatogrel, cenerimod, which are starting phase 3 or already started phase 3, more in specialty markets, a selective orexin receptor 1 antagonist, and early products. So the second priority of our key strategic priorities is make money, get the company to a profitable level, because otherwise we will not remain independent. How do we want to make money? Daridorexant already mentioned. This is in the primary care market, so we are preparing for a successful launch. Clazosentan, also mentioned, and more in the specialty field, cenerimod for lupus and selatogrel for acute myocardial infarction. On the other hand, we also have some aspects of earlier research that we are collaborating mainly with Johnson & Johnson on ponesimod, which is already on the market. So we have a small royalty stream coming from there, aprocitentan, that should hopefully soon come. A T-type calcium channel blocker that we are developing in collaboration with Neurocrine Bioscience in the field of juvenile epilepsy and essential tremor, and vamorolone, where we are kind of an initiator, and now it's in the hands of Santhera and Reveragen. Something got messed up, so this would have been the first slide. So we discussed number 1 and number 2. So no, actually we discussed number 1 and number 3. So you see we have 5 strategic priorities. Number 2 would be build a world-class commercial organization, which is obvious from the situation we are in. And then extremely important, in order to get to long-term success, we need to fuel our pipeline with new discoveries. And for that, we want to utilize state-of-the-art technologies. That's why probably also I'm here today. So coming to priority 2, build a world-class commercial organization. I'm not going into the detail, but you can see we are present in the US, we have a presence in Europe and Canada, and we have presence in Japan for the moment. And in Europe, we have these main markets like Germany, Spain, Italy, France, and the UK, where we start being actively present. That was actually slide number 1. It's full of surprises, you know, coming home and everything is full of surprises. Cool. Okay, so actually I wanted to start to tell you a little bit about a certain philosophy that we had at Idorsia. So I'm just going to adapt according to how the slides pop up. You may know, maybe not exactly the number, but we have just in this direction in 2 big things, more than 550 lab workplaces where people are performing lab work. We still have a strong balance sheet at the moment, so we still have some money to spend to develop the company further. We have, what I already showed you, 11 compounds in the pipeline, many of them late stage. And global commercial, I already showed you, and we have approximately 1,200 people now working for the company. Probably, as of today, it's even more, taking the whole commercial organization, which is built up into account. Okay, now we are with priority 5. Utilize state-of-the-art technologies to drive our innovation. So I would focus here only on drug discovery. Here, I think our main focus in modern or in novel technologies is we want to implement artificial intelligence machine learning, which requires data production. We are having a strong team of computer modelers and electron diffraction, hopefully in the future, which will move us forward. So I think now is going to come priority 4. Yes, so this should have been before 5, because I think it's very important to understand 5 that you see 4. So we have decided strategically that we focus on small molecules because of the following reasons. Small molecules are suitable for acute as well as chronic treatment. They are suitable for oral use, so it's very convenient for the patient. And very often you have a very clear patent protection. So you know either I have the protection or you know I have a problem. This is more complex with biologics. So that's why we are very interested in organic chemistry. So we have a lot of organic chemists. I think I have approximately 140 people on site working in synthetic organic chemistry. That's why we are very interested in getting connected with top quality synthetic organic chemists, because from time to time we need to hire one or the other in order to have newest things up and running. And the other aspect already mentioned would actually be priority 5. Okay, now I think that was expected. Very quickly, how do we work? So we usually think about new targets, biologically relevant receptors or enzymes, or let's say, yes, biologically or medically relevant targets. This requires very often a lot of literature work, then confirmatory work, and once we have confirmed the target, we start generating results with so-called high-throughput screenings, whether we can activate or inhibit or modulate the target in the way we want to modulate it. Once we have sorted that out, we start to analyze the data we generated, and then we create hypotheses on where do we want to go with the starting points. So we design compounds, and then the synthetic chemist comes into play. We synthesize the compounds, we test them, and we input the results back into the design cycle. So this is a constant running. Sometimes, I think for daridorexant, if you take molecule by molecule, we took 25,000 cycles until we selected a compound that we call preclinical candidate for preclinical development. So this means we characterize the compound up to the level that afterwards we are allowed to do first human clinical trials. Drug design, drug discovery. You may have heard about the lock and key principle. So you could actually consider small molecules as keys and your relevant target, your receptor, as a lock. So you design, that's also not exactly the right order, I think. You design your keys and then you test whether they fit into the lock. Here you can see that your key fits into the lock, but it's not optimal because you have free space where the surface is black. Okay. It's getting exciting. So okay, we changed the topic. You may have understood the other one. So you will have keys which do not fit. Then you get inactive results in your screening. And then once you understand your system in 3 dimensions and with biology, you will obtain a key that perfectly fits. And then you can either block the lock or open the lock or play with the lock when you have the key. So you can actually imagine that we, as medicinal chemists, what we are doing every day, every week, we are looking for the keys that perfectly fit into the lock. And this is heavily affected by 3D thinking and 3D structure. So because flat only on paper, in reality our world is 3-dimensional. So our small molecules have a shape and our receptors have a shape and the binding pockets in the receptor have a shape. And what is extremely important, all these shapes are dynamic. So they can adapt, they can change, they can show up in different conformations. Then an additional aspect which is extremely important is chirality. So nature is extremely intensely chiral and small molecules can also be chiral. Sometimes it can be an advantage if you have a chiral molecule because it binds better to your receptor. For the chemistry aspect, an achiral molecule would sometimes be more advantageous. And all these efforts on protein structures and on small molecules help us to understand, if we combine them, our structure-activity relationship in 3 dimensions, which is extremely important for the modelers to efficiently design the next round of compounds and to speed up the projects. Okay, I think, let's see. That's the key which doesn't fit. You see there is a part which is wrongly designed. And So what we are doing is looking for the green keys which perfectly fit into the lock. So here I just want to show you in a different perspective the interaction of daridorexant bound to the orexin-1 receptor. This is the space fill model. You can see that actually the space fill or the compound nicely fills a large part of the binding pocket. We very often also look at it in the ball and stick model in order to see it a bit better from the chemistry perspective, but this is much less intuitive to see how nicely the pocket is filled. Now let's see whether I can go to, no, where is the most important slide here? Is it at the end? Yes. So here I would just maybe like to show you how daridorexant, you can imagine how daridorexant interacts with its receptors. So this is a small animation that Naomi generated for me. So you can see it goes in, the binding pocket is on top of the receptor, it fits very nicely. And that's actually what I personally use from what all you as experts generate. So I love these things. So now let's see whether I can get back to just quickly. So I think we were here. I think we went into the importance of 3D in all aspects. Experimental structures of what I just showed you from daridorexant with its receptor are not always easy to get, or sometimes you don't even get them experimentally, you need to model them. In order to improve your models, because this is challenging and there are several unknown aspects, you need to understand the conformers of the protein, you need to understand your small molecule, you need to understand the stereochemistry and the binding site. And where hopefully electron diffraction in the future can help is in the two aspects which are pointed out in red here, so that we get more information about the small molecule and more information also with respect to stereochemistry of the small molecule. So let's quickly change topic, drug development. So why are 3D structures also important in drug development? If you want to submit a dossier of a molecule that you would like to get approved as a drug, you need to fully understand its three-dimensional structure, you need to confirm its chirality, you need to confirm its composition. So here it's very important that we get this information, and the most efficient way is also to have an X-ray. And then I think something which is actually inverse, and where we really hope that the electron diffraction will be able to help us, is that you can characterize the solid states from single crystal data. And this is really something where we are very keen on learning how well this works with electron diffraction. Very quickly, what we do on a level of crystallization, we have approximately 1.6 structure requests at our company per month. So this means approximately 20 structures that we solve per year. The reasons for it are absolute configuration. Then what I just pointed out is simulated powder patterns to analyze crystal forms, confirmation of chemical structure or confirmation analysis. And approximately 50% we are not able to crystallize them very easily, so then our service companies need to invest into crystallization. Success rate for the first part is 60% in 4 years. For configuration of the absolute structure is also 60%. And for the simulated powder patterns, I think there we have really to invest a lot of work until we get it by traditional methods, and there we really hope that electron diffraction will change the world for us. And for the aspect of where we need crystallization help, approximately 50% are successful and 50% fail. And we really hope that with the potential that ED shows, we will kind of reduce the failures to an absolute minimum and get the information that we are missing now. So I think this we discussed, this we discussed. And then another short aspect on why perform solid state characterization. Organic molecules can crystallize in different forms, so-called polymorphs, which can also include hydrates or solvates. And polymorphs usually have different properties. So here it's mentioned different thermodynamic stability. This very often results in different in vivo behavior. So that can change completely the pharmacokinetic properties of the medication if you go from one polymorph to another polymorph. And it's extremely important that you are absolutely clear on these aspects before you put the medication into humans, because if something happens in the pill with respect to polymorphism, you can create a completely different readout as you would have expected based on your initial aspect. So that's really absolutely key for industry. And we also hope that in this aspect, electron diffraction will help us to speed up the process, the accuracy, and the guarantee that we always know what we are doing at the end. And with that, I think, sorry for the kind of a bit difficult flow of the presentation, but I would like to thank Naomi for her help in the preparation of the slides, then certainly Martin and Jean-Paul Clausel for the enormous opportunity they created with Idorsia, and you for your kind attention. And just to be clear, the mess in the slides is certainly not Naomi's fault.
No, you coped extremely well with that. Well, I don't have to congratulate you with that, but no, you seem very flexible, very adaptive, and yeah, I now understand how Idorsia became such a successful company. Yeah, it seems trivial to copy a slide set into another one, but somehow we managed to. Sorry for that. Sorry.
Thank you very much.
Yeah. Next speaker is myself. There we go. Yeah, I think it was in fact a great talk of Christoph, which touched on many points and applications and advantages of electron diffraction already. Kind of in parallel, we did a survey to get an overview. What could be the use of electron diffraction? Where do we stand? Where are we going? And that's what I wanted to present briefly in the next 10 minutes. Yeah, key performance indicators. In fact, we heard many of those already. The ultimate KPI of electron diffraction, obviously, the fact that you can get a structure from a nano-sized crystal, which would not be possible for X-ray. So it's a matter of fact getting an X-ray, getting an atomic structure, or not getting a structure, which is the difference between X-ray diffraction and electron diffraction. But there are many things, and we just heard them. You can do crystal mapping, which is a bit difficult with powder XRD, for example, or other XRD technologies. You can do crystal by crystal, go through your sample and analyze each of them individual by crystal mapping. Even you can detect crystallinity within a seemingly amorphous sample. That's often a tedious or difficult task in powder XRD, where you get a big hump and perhaps some tiny spots from crystallites. And yeah, the thing that in your electron microscope you have almost the bulk of your sample, but at the same time the bulk is separated into individual crystals, gives you the opportunity to investigate those individual particles, which is tedious or difficult or impossible if you're looking at a real bulk sample with only one big X-ray beam, and getting similar information out with XRD is complicated. Yeah, why electron diffraction now? Technologies indeed come together. We are profiting from novel detector technology. We're taking advantage of better electron sources. We have seen what's going on, but yeah, we definitely missed from the very beginning the crystallographic approach or possibilities or flexibility that some of us from our founders and within the company knew from our crystallographic background. Within the application of electron diffraction as it was established in an electron microscope. And so I think the big come together, the marriage of electron diffraction as it is and was typically used in electron microscopes, plus the experience and knowledge from X-ray diffraction, putting those two together, I think that's the big advantage that we will see as of now. And bringing these technologies together brings electron diffraction, we think, a huge step forward. Yeah, and at the same time, bringing these technologies together, we see also a huge evolvement in terminology. We have gone through many acronyms, ADP, ERED, I can hardly remember them. But luckily we are converging now to I hope at least that 3D-ED will continue as a terminology for electron diffraction as a technology. Yeah, well, no, that was the wrong button, sorry. I was already looking forward to another surprise, but who knows, perhaps I also get surprises. Yeah, so as I said, we did a survey, and I won't go through the numbers, but yeah, we consider it as a quite representative survey. We had over 100 respondents, and we had a bunch of 26 questions, and in that sense, I want to present one or two of the outcomes of that survey, which gives us, as a company, but possibly also you as a user, an insight on what is the feeling about electron diffraction in the field. Yeah, we see that many respondents did not have access to electron diffraction. That's, yeah, understandable. There are not many electron microscopes around, but more people could have access to electron microscopes, but they don't use this facility, or it is difficult to access them. We know the hassle in optimizing an electron microscope for diffraction. So yeah, I think we can understand that the access is not easy to this technology. At the same time, many of the respondents were not trained in the technology. That's also kind of a hurdle in doing electron diffraction experiments. And yeah, we have a few specialists in the field among the auditorium, and they used much of their career in getting specialists in this technology and getting good electron diffraction data out. At the same time, we see that only 10% tells us that they don't have applications. In reverse, one could say one should conclude that more than 90% of the responses do have applications for electron diffraction. Yet at the same time, they don't have any access to instruments. So yeah, I think that's a good indication for us as a company to continue along the lines that we have started and make systems that are more accessible and would apply to all the applications that people think of. And even more clear that 98% of the respondents do see an advantage to use electron diffraction for their samples, but one needs access. Right. It seems clear that the community that replied sees electron diffraction as a necessary tool. It's a relevant method to be used within crystallography, within structure determination. But there are some hesitations. Poor quality, for example, quality criteria not met, incorrect structures. And again, there we use that as a stimulus. We as crystallographers had similar experience, of course, mainly Gunther and Gustavo doing this kind of work in practice and running into the limitations of electron diffraction and experiencing these mediocre data quality in converting that in a machine that would be able to get you better quality data. Opinions on electron diffraction, it's a proven technology. That's good to hear, but it's not widely applied. Very few thought that electron diffraction will never reach the required accuracy. Also, that is a very promising outcome that that gives us the faith to continue with our equipment and available instrumentation too complex. That's something we are working on, making it more easy to use. And there's no instrumentation at all. Well, that's only very few replied that there is no instrumentation. Thus, in general, there is instrumentation, but we have seen it's too expensive, it's too complex. You need to be trained, and all these issues need to be addressed. And we are working hard on in fact addressing all of these items and making electron diffraction as a technology more accessible, more approachable, more easy to use, and providing also more accurate data. Now, it's not only structures, of course, that one should think of. That was even the primary target within our company, but we soon figured out, also thanks to Danny, our sales manager, with a lot of experience in the pharmaceutical field, that that is quickly kind of a range of applications that we can incorporate in our equipment, that we can tackle with the electron diffractometer. And also the respondents saw these applications. Working with phase mixtures is a great application for electron diffraction. Looking at twinning or crystals too small, of course, and all of that can be extended even into EPDF. Crystal mapping, as I said, degree of crystallinity, quality control, fraud detection, and IP issues that we can discuss today. Even fragment screening or forensic applications would come into play with an easy-to-use, accessible electron diffractometer. And with that, I would like to hand over and start our panel discussion. And that will be moderated by Sebastian Scheck.
Why we have invited also, and I'm very glad that you all followed our invitation, and really appreciate and also thank you in the name of ELDICO that you came to the beautiful town of Basel for this evening. We have asked them to come to here and to join us, to share their different perspectives on this new technology with us. Because there is not only, we will have some guys more on theory, on modeling, on electron density from this part, but we also have an array to the complete other part for the practical thing, how to get, at the end of the day, structures out, which can then help us in our research endeavor further on. We will have always, and that is very fruitful, a dualism between, on one side, organic chemistry, maybe even solid state physics, and what I would like to call industrial applications. And a lot of what is with ED is in this area. But coming to Basel on the other side is, of course, also very much about life science and pharma and the pharma applications. And we are happy that Christoph shared already a lot of insights with us on this part. And we will have the dualism, let me say, we are starting now, and Hans Florian told us, hey, you are now the nucleus. But of course, we are thinking bigger. And at the end of our panel, we will have an outlook where we think also about future applications and what ED could do in 5 or 10 years for us. So we will structure a little bit for the housekeeping. We will structure the panel in 2 sections. The first section will be that each of you, we have talked a little bit about that. We give a lot of 5 minutes statement about your experiences with ED. Of course, first about yourself, where you're coming from, Also, let me say what the work is what you are involved currently, and then going over to the perspective you may have with the novel technology. So that will be the first part, around about 5 minutes each, so around about 30 minutes. Please excuse me if I will try to manage also that in more or less half an hour we will have finished that, because we are very eager to get also your questions. And also, when I say your questions here in the audience, in the room, as well as from online, the online guys, they have the privilege compared to us that they can type in already a lot of questions, whereas we can't. We have to memorize them. And so we have to memorize them and then ask them in the second section. So let me start with the panel. I would like to introduce, let me say, with 2 sentences each of our panelists here, and then I will hand over to Michael for the first part. Michael, indeed, is Michael Hennig. He is the CEO of LEADEX Pro, and startup from the Paul Scherrer Institute, and he's working on the X-ray, but also electron microscope structure of mostly membrane proteins. So he is the guy in life science who describes the shell for us, as if I may come back to the metaphor from Christoph, where he showed us the protein around. So, pardon me? The lock. You are working with a lock. Now, I'm coming to Paulina, and she's actually from University of Warsaw. Complete different angle in the way that she's coming from modeling electron density, and she's doing this with peptides. And I don't say more. You will tell us then again more. I'm coming to Bernd Hinrichsen. He's joining us from BASF, and he's the head of the structural research lab of BASF. He's trained and coming from the powder X-ray diffraction. It will give us a perspective what his hopes are for electron diffraction as well. I'm coming to Michaela Popp. She joined us from far away, from Cluj in Romania, actually from Transylvania, as I learned.
Very interesting story.
She's joining us she's the Chief Scientific Officer of TerraCrystal. So she's coming from the very practical side, really getting first crystals out before then these guys can eventually do a structure. And Christoph shared already with us, it is quite difficult sometimes to get sufficient crystals. Okay, coming to Christian Lehmann. He is the head of crystallography and electron microscopy of Max-Planck-Institut für Kohleforschung in Mülheim. For those in the audience who do not know, this MP is very famous for decades-long work in organic chemistry. I may say so. And and of course, he will also share with us then his perspectives working on organic compounds. And then finally, and this is also very important for us, the patent aspect. So we have Michael Kaufmann with us from patent lawyers Bohest, who will give us at the end the perspective from the patent lawyer, maybe also on what he has heard before in the audience and in this panel. So, sorry, I'm always talking too much and too long, and don't want to deprive you from time. I may hand over to Michael, and so your perspectives.
Yeah. First, a big thank to you for this great introduction. I think there's almost nothing to tell anymore. So you have really introduced ourselves, all the panel, I think, very well. So my name is Michael Hennig. So I would call myself a structural biologist, and I have worked in pharmaceutical industry for many years, more than 20 years with Roche. And since 2015 for the company LEADEX Pro. So the company LeadExpro is dedicated to the structure determination of membrane proteins, so to enable structure-based drug discovery for membrane proteins. And membrane proteins are very challenging. You think about GPCRs, ion channels, transporters. They are integrated into the cells and they need to be solubilized first and then purified and then used as proteins, as samples for structure determination together with mainly small molecules. We also work from time to time with antibodies, nanobodies, and other biopharma compounds. However, the dedication is indeed small molecules and very much like that was introduced by Christoph Boss earlier. And so we do the complex between the key and the lock, and to better understand the fit of the key to the lock. So that is perfectly that what we are doing all the time. Maybe a small anecdote about the company history of LeadExpro. As I said, co-founded in 2015. And at this time, X-ray crystallography was a key technology to drive structure determination. And this, of course, was very challenging because crystallization of membrane proteins is very challenging to grow good, well-diffracting crystals for X-ray diffraction. But this was the key technology at the time. And I remember my first board meeting. I was sitting then together with the board and said, you know, X-ray crystallography is my passion, yes. However, there is a new technology on the horizon, and this is called electron microscopy. So to work really with electron microscopy, with electron microscopes, to visualize the structures of targets and so on. And of course, this was a bit of a shock to the board because they said we focus only on X-ray crystallography and not anything else. However, I said that is really an important development and we should follow it. And today, I have to say, I would say 80% of all our structures we do with electron microscopy and only 20% with X-ray crystallography. Mainly the GPCRs are left for X-ray crystallography. The rest is done with electron microscopy. And a similar development I see for electron diffraction. Here at the moment, this is still early and it might take a bit more development time to really be feasible for membrane proteins and the structure determination. However, I also here see a great future that more and more targets will be amendable for structure determination with electron diffraction. From this point of view, I like this area a lot and monitor the developments, and I'm looking forward to also have electron diffraction in a portfolio of methods, technologies to drive structure-based drug discovery.
Perfect. I appreciate very much. And you know, also you gave us already the mood, so to speak. Go on, guys. Go on, guys. So to make it a little more interesting, you are not talking in this. You are not talking in this order, but we are mixing also to challenge the guy on the video actually. So we wanted to continue with Christian actually, and there's one idea behind. You know, we have, as I said, there is guys coming from structural biology, you know, all the time. So, but we want to have the dual role. So we want to give also organic chemistry and the solid state physics also a kind of a voice. And so the floor is yours, Christian.
Hey, thank you very much, Sebastian. Well, a few words about myself. Just calculating, I've been in X-ray crystallography for about 35 years now. And most of that time was really doing service crystallography, helping the chemists to know what their structures look like, what their molecules look like. For the last 14 years, I've also been in charge of electron microscopy in our institute. So I'm in a kind of position where I can really combine the two technologies to hopefully put them to a better end. We have been trying to help our customers by getting to smaller and providing more and more X-ray intensities so that they can get answers faster. But what they do is they bring me smaller crystals. So it's a battle I keep losing all the time. We even have access to a synchrotron beamline. And so with electron diffraction, 3D electron diffraction, I can foresee that eventually we will win the battle. So, because if they get any smaller than what you can put into the electron microscope, it won't be any more crystalline. So that, I think, is a great perspective. But on a more serious side, we are not only looking at organic molecules. The Max Planck Institute, you might have heard or noticed last October now with asymmetric organocatalysis. We have earned our second Nobel Prize in 60 years, but originally it was on polymer science. And this was something where organometallic chemistry played an important role. So this still is the scope of the compounds we look at. So as a chemical crystallographer, which I think I can call myself, I really take great interest in different molecules and the structures of different molecules and understanding the chemistry and understanding the chemical bonds in that. We also take this sometimes a little bit further to look at, and I think Paulina will talk much more about that, about the real electron density, not just kind of approximating atoms. But I, for myself, really foresee that in some time to come, 3D electron diffraction will really not probably take over. If you have very good crystals, I think X-ray diffraction does have some real potential and has some reason to be there, because we do fight some physical problems if we diffract electrons. And I know there are brilliant people around who kind of overcome what is now called the R-value gap. I think that will kind of shrink as we progress in the next couple of years. But for many things, electron diffraction, both in the world of small organic molecules, in organometallics, in inorganics, and in zeolites, we have seen many applications of electron diffraction over the last 10 years, 15 years already. And so I'm very much looking forward to put my hands on a machine which will really be able to do what I do in an X-ray diffractometer, put a crystal there, press a couple of buttons, and then get a diffraction pattern to have the same thing in an instrument which uses electrons instead of X-rays.
I appreciate very much. And again, Eric, you got the boot, yeah? You just 3-4 buttons, not more. 3-4 buttons, not more. So, okay, and we now want to hand over to Michaela. And again, it's a little bit of a difference. And now comes the practitioner.
Yes.
Yes.
With hands-on.
Exactly. So share it with us.
Well, yes, yeah. Well, actually, I'm glad that I have this occasion, and thank you for this invitation. My name is Michaela Pop. I'm the scientific lead of TerraCrystal, Transylvanian CRO. I will promote it from now on. So basically, we provide full solid-state crystallization and polymorphism services for pharma industry. This is our main focus, and because our projects are quite, we strive for scientific solutions to our customers. We are continuously looking for advanced methods to characterize our crystals and powders coming from a crystallization screen. Well, in that respect, crystal structure determination is very important. Why? Because knowing the arrangement of the atoms in a structure will enable basically to understand its function and how it performs in the body. I think the first presentation was very good in highlighting exactly in the drug, in the drug development how important is to know crystal structures. So in our projects for customers, we want to provide the best solutions to polymorphism. And we use actually information from crystal structures when we want to confirm a certain polymorphic form, if it is a polymorph of hydrate, co-crystal, or a salt. So ultimate proof is to have the crystal structure, but also to understand the relationships between the polymorphs, and also to predict the properties, basically to understand the processability issues that can occur, the compressibility and so forth. So basically to select the best form that we can recommend to the customer to proceed in the development. So not only for the lead compounds, as we saw in the presentation, that we look for these keys, but the most important keys, we look how they connect in a crystal structure, and we calculate the energies of a crystalline structure to see what is the stability, what are the issues, is there space in the structure, is solvent getting in there, for instance. So that's for importance of the crystal structure, especially also for the polymorphism. But so what we do, we have in many cases crystals in a screen when we look for polymorphs, but in many cases also not large enough crystals. And we heard about this problem. I think from that perspective, electron diffractions is amazing, and it would bring a huge added value to the screens if we could determine the structures of each and every form, and especially the stable ones, the important ones. And then, of course, we could also make a better selection and we could avoid a lot of problems. So from that point of view, I was searching for possibilities to explore electron diffraction, and I had the chance to work with ELDICO on a pharmaceutical compound, and I was really amazed because I tried before using electron microscope, and I was able to solve the structure, but the modeling it was a combination, let's say, between single crystal. So just the indexing was done by microscope, electron diffraction under microscope, and then we needed to combine with simulated annealing. But with ELDICO, with a dedicated instrument, I was really amazed, and I was a bit skeptical in the beginning, I have to say that. But so I wanted to get the data and to do the crystal determination myself, and I was really amazed that ab initio, a pharmaceutical compound, and not a simple one, because it was even with Z2. So it was. I was really enthusiastic and amazed that this worked so easy. So well, it doesn't say that it will work for everything, but I think it is worth trying for each and every. If I would recommend the customers and the Dorsia, and we had a good discussion earlier also with Marcus. It will bring an added value, and it will in the future, I think it will increase the output of crystal structures which are determined. And as you know, this will go in, hopefully, in some databases, in CSD. This will enable then, it will be like a snowball modeling, better designs of the screens, better crystal structures, and fast time to market, I would say. So this is how I see it in the picture. Really a catalyzer of bringing drugs faster to the market.
Perfect. I appreciate so much. And yeah, I thought you are already filling up a little bit your prophecy where you said at the beginning, 2015, you had it electron microscopy almost zero, and now he is with 80%. So I just combine that. So that would be good. Coming to Paulina, going back a little bit to the theory, if I may say so, yeah, and to the electron density, but at the end of the day, that's what we're measuring with electron diffraction.
Yes, so I'm from the University of Warsaw. I consider myself a chemist, or maybe crystallographer, or even a quantum crystallographer. So I started with X-ray diffraction, and I actually haven't stopped at the three-dimensional structures of atoms and atomic positions. I just wanted to go further. So what we were doing, we were trying to obtain electron density distribution in a quantitative way from X-ray data. That was a really very tedious way, so I moved slowly into modeling. We are constructing electron density models which are designed to fill the gap between quantum chemistry and molecular mechanics. So they are to be much more accurate than any molecular mechanics or force fields can give you, but much faster and easier to be used than quantum mechanics. So the model we're focusing to reconstruct electron density for proteins or protein interacting with small molecules. So exactly to start from three-dimensional structures to reconstruct electron density, and then to understand why this key really fits into the log, because it's not about the shape, it's also about interactions. So we try to describe those interactions in a more accurate way, not only on the basis of geometry. But the same models which you can use to characterize interactions, you can also use to interpret diffraction data. So we are going back to the diffraction, to the crystallography, because, as you said, what we are measuring by X-rays is electron density, and by electron diffraction is electrostatic potential, which is also dependent on electron density. So the models we are developing, we are using them now to extract more information from your experimental data, from diffraction data. So we will getting much more accurate three-dimensional structures, but we have also access to accurate electron density or electrostatic potential descriptions. So for me, you really don't have to convince me that electron diffraction is giving you three-dimensional atomic structures. That's obvious. I want to go further. And I really would love to extract from electron diffraction data electrostatic potential in a more accurate way to be used to describe interactions on the basis of that. So I think that's the future, and so I'm looking further. And this is, you were focusing on small molecules, but I think this will be also very important for macromolecules and complexes, and to see really the true interactions between them and to characterize them. So that's a next step to tackle.
Great.
Thank you.
Appreciate very much. And indeed, yeah, the interaction between the lock and the key. And as you said, there's more than just the electron density. There's really something going. There's a kind of chemistry going on, and will be highly interesting. And I'm looking forward also for the next years and of all your research. Maybe also with Michael together. Coming to the last speaker of this evening, we learned already that the ED is a very novel field. But I learned in my career, even if it is very early, go out and reach for the patent lawyer in your company, in your organization, wherever, because usually in the early times you're spreading, you're sharing your knowledge, or you're doing all this, and you may lose opportunities also for intellectual property protection. And so this is why we also want to focus in this session a little bit about the patent protection. We do this in two ways. The first one, we would like to give Michael to present the patent lawyers, the law firm he is representing, and a little bit about himself and how he comes into this field. And he may help us later also to sort out if questions come in terms of the patents. Michael, it's yours.
First of all, thank you, Eric, for giving me the occasion to be present at this meeting. My name is Michael Kaufmann. I'm a patent attorney. I'm working with a company in Basel. The name is Bohest AG. We are a very old patent attorney firm. We have been founded in 1888. We're one of the first patent attorney companies in Switzerland. We were founded the same year when the Swiss patent law was established. And I remember there was a debate among the first patent attorney companies in Switzerland who would get patent number 1. I don't know which one of the companies won that competition. But we have now a main office in Basel. We have a branch in Buchs. We are now 8 patent attorneys plus administrative staff. So in total, we're about 25 people. For Swiss standards, we are a large company. But in comparison to, let's say, United States or Chinese companies, we are minute. I have to admit that. We do all the aspects of patenting. That is, we get patents granted from filing, prosecution, grant. We also defend patents. You can imagine that not everybody is happy with being faced with a competitor's patent. And there are possibilities of getting rid of a patent by opposition or by nullity action in court. We do that as well. We fight third-party patents, and we defend our own clients' patents against third parties. And then there is a third important aspect that we also do. If you would like to go to the market with your product, then you must beforehand be aware of the patents of third parties that you might infringe. And that's also part of our job. We have to analyze the product that is about to be placed on the market. We make a search for the third party rights that might be relevant. And we analyze and we give an opinion to the client. Your product is infringing that or that or that patent, or it's free. And that's an important assessment for your client. Now, my personal background. I'm organic synthetic chemist, but that was a long time ago, about 30 years, when I made my PhD in the late 80s. And then I went into Australia for a postdoctoral study with Professor Athelstan Beckwith at the Australian National Research University. And that was for radical chemistry, which at the time was a hot topic. I think it's no longer the case now, I don't know. And I also did, during my graduate studies, an introductory class in X-ray analysis with Professor Bergi in the University in Bern 30 years ago. And there was a small publication that arose out of this study. So I have very rudimentary knowledge about X-ray analysis. I have to say, very rudimentary analysis. And I have been in the patent business for about 25 years now. So we have a certain experience now. In our firm, we do all the fields. We do organic chemistry, synthesis, mechanics, electronics, computer science, and so on. We do all the fields of patents. Of course, my field is organic patents, medicine, pharmaceuticals, foodstuffs, formulation, and so on. Now, about the second point that was given in the document. Of course, we don't do any research of our own as a patent attorney's firm, neither for ourselves nor for customers. But our primary job is to translate the research that you do into patentable patent claims. And I don't know if all of you are aware of the significance of a patent and a patent claim. The patent claim is the most important part of a patent. That defines what the scope of protection is. Imagine in the old days in the gold digging field in the United States, the Wild West, there you also had a claim, and that was your territory. And you said, no one else is allowed to dig here for gold. That's my territory. And that's very similar to what a patent claim is. That's your territory, and you are saying to the public, no one else is allowed into that territory unless I consent to it. So a patent claim has to be as broad as possible. It should cover in particular everything that you have made in the experiment. It should furthermore be broad enough that no competitor can bypass what you have done. So it should be considerably broader than the experiments that you have made. But on the other hand, a patent claim must be novel and inventive over the prior art. So it cannot be arbitrarily broad. And that's in fact one of our key jobs. Make the patent claim as broad as possible to cover everything what you did, all the possible equivalents to what you possibly did, and on the other hand, still be patentable over the prior art. That's our key task that we are doing as patent attorneys. And then about the expectation that I can have in electron diffraction. I hope that it will become a reasonable tool in identifying compounds in patenting for both drafting patent claims and also for analyzing competitors' products, whether they infringe your patent. And I have here on the stick a few examples of patent claims, how they could be drafted with diffraction features. But it's a PowerPoint presentation. I cannot show it right now. Yeah.
Okay, but we are sorry. We are very sorry for that.
Yeah.
But definitely, but maybe you can share also in over the upper row. If we want to have it a vivid. We want to have it a vivid example over here. But I appreciated very much also your second part. I recalled back to all the companies I were working for, because it was exactly the melody what you said, which all the patent lawyers in the companies of our guests, if I may say so, would probably have said the same way. Exactly. And always, and every day, and they say it every day again. And they say it.
So my discussion is a little bit short because I cannot say very much about the third point. That's in fact, you have to tell me what you expect.
Exactly. We have to work on the next couple of years, and then we're coming back to you again, and then we're coming. Okay, so that will be for the first part of our panel session. We are a little bit over time. I hope that also the online audience has still, let me say, enough energy to listen now. And we would like to open to the audience for questions as well as to questions from the online panel.
If you start to do this kind of job in your instrument and you have a tablet, how much time do you need to find all crystals? What is your...
Well, we would need probably 30 seconds to move from one crystal to the other, and then if you would require a complete data set, and to be sure you would need a complete data set, that would require a minute of measurement time. So in passing through a grid, you can do 40, 50 crystals an hour if you would automate and make the system more adept for that. Yeah.
Let me add a comment to what you were saying. And so basically, you don't need to get a structure. You need to get just a unit cell. So it would even be enough to have few degrees of rotation to get a unit cell. And once you have a unit cell, you have a powder pattern that you can simulate. You can compare. You can say, this is polymorph A, B, C, D, and this is a new polymorph, or this is a polymorph that it's infringing my patent. So in that sense, electron diffraction would be a game changer. And I think importance is the IP community needs to recognize the potential of this technique. Once the IP community recognizes this and it's established as a standard procedure to publish or to protect your materials, then it would be a game changer for sure.
Okay. Also, as your moderator, I have to think a little bit about the broad audience what we have here. There's some very special guys who have already deep insight into ED, into crystallography, and so on. Others may come more from let's say, from the research side, but on the use of these data only. I would like to start, let me say, with a question to the audience here. Also a little bit to train the audience, honestly, in the way. What is the crystallographer's point of view on your specific expectation? What would you do tomorrow and let's say in a few weeks or years with ED? Let us say, is it about polymorphism? Is it about co-crystallization? Is it about what size of the crystals you usually? If somebody would like to, it feels encouraged to give a few points, let me say, for the audience to understand what the nice lady was talking and Paolo was talking already about here. Who feels encouraged to give this seminar for? Is it you, Bernd?
Well, I'm going to say something completely different because. Okay. Oh, sorry. Of course, according to your question, but I think I'm not into the pharmaceutical part. I'm going to say disorder because I think disorder or the aberration from the perfect crystallinity is something that is profoundly important for the materials that we use in our everyday. And understanding that is something that is really going to change the way we can redesign and produce better and newer materials. And that is exactly sort of the break of the crystallographic order. That's just something that interests me personally very, very much. And I think this is where we're going to see a huge development in the years to come, especially with electron diffraction, because it's so powerful with small materials.
It's a significant answer to my question, nevertheless. As you said, it is not look at the perfect crystal, not look at the big thing, look at the disorder, That's what we're taking home. But Michaela, would you like to answer my question also a little bit?
What I would do? Well, I think I don't know if I stressed it enough, but I would definitely determine the structure of everything I find. So each and every form, either it's co-crystal or solvate, or and it can be that it has disorder, of course, or not. Even though, in my previous experience, sometimes you have a pattern. I'm also, by the way, by education, a powder diffractometrist. Sometimes you think, well, I have a three-dimensional structure because you see a pattern with more than 15 peaks, and then when you look with electron diffraction, you think, no. Talking about disorder, it's a mesophase. So the structure is not three-dimensional maybe, and you will be able to find that out with electron diffraction. So molecules become complicated. You need this very, with powder diffraction, you look at the bulk, but then you need to go to zoom in. What exactly is in that crystal? Is that disorder? Not. Is the structure perfect or not? This will help you later on to understand how the molecule likes to crystallize. And well, I think I have the advantage because we get a lot of samples. We see crystals smaller, bigger, beautiful, not beautiful, red, green. So I would just determine every structure.
Give me a very short figure. How big are your crystals?
Well, of course, with single crystal, we start thinking of single crystal X-ray diffraction if they are more than 30 micron in 2 dimensions.
Okay,
at least, yes. So, but usually they are not, of course, when we do precipitation techniques and so forth. And we spend months to get them even sometimes to 50 micron or 100 micron.
Okay.
Yeah.
Some question from the audience. A little bit in that way, as you said it also, or let me say, what is about polymorphism? You know, I'm coming to your disorder because this gentleman here is very much concerned about the arms race, what electron diffraction may even also fire in for the polymorphism and the litigations coming around polymorphism. Somebody an idea about that?
There is a decision from the late 90s, and they have said the simple fact that you provide a further polymorph without having any surprising advantage for that polymorph is not inventive. So by simply creating a new polymorph, you don't get a patentable invention. Unless if you have for that polymorph a surprising effect like a favorable dissolution profile, so then you can get a patent for that. That's inventive. But the simple provision of a polymorph as such is not patentable.
Consequently, if this is just an impurity and is not a major part of your galenical formulation, I don't think that is a big issue. That would be my judgment.
Michaela?
Has to do how many crystals of the other polymorph you find in that tablet, taking the example he gave. If you only find one, which is different, they will say, well, with the state-of-the-art method, which is X-ray powder diffraction, is not detectable. So no infringement, I think.
Yeah. That would be my...
Yeah.
Also a question to Erik, but also to you. How are you dealing with crystal water? You know, because is crystal water an impurity in your sense? Because crystal water, I mean, apparently, is a major part of to describe any kind of crystal structure.
Crystal water would be a challenge in the vacuum. Most cases, the water will disappear out of the crystal. In many cases, the crystal would collapse, so then you don't have the information that you are after. So you should avoid that. And the obvious way is to go to low temperature, to keep the crystal at liquid nitrogen temperature, for example. Then you would slow down this process considerably. It's not a 100% guarantee, probably, but we are also thinking of different approaches to that problem. There has been publications on using a kind of double grid, a sandwich grid, where in between you can put your crystal even with a little bit of solvent to keep it in the mother liquor, to keep the crystal in its natural state, and in covering these grids, which normally only have some carbon, but you could cover them with graphene, which is a lot more robust against the vacuum. You could kind of seal your substance from the vacuum, and that's the thing we are about to investigate. It's not completely done, but we have all the material in house to look into this. That looks as a promising alternative for those which don't have low temperature attachment, but obviously there will be a low temperature attachment to the system that we have been starting working on.
Are you aware of azithromycin, this macrolide antibiotic? That's an old example. And this did not crystallize well as such, or even as the monohydride, so it was difficult to get pure. And then Pfizer, by coincidence, found that in the presence of water, this azithromycin would crystallize nicely as a dihydrate. And that was a very good patent. And it has been cited many times, and they claimed it explicitly. Crystalline azithromycin dihydrate, that's it. A super claim, valid. And the difference was in fact only that there were 2 crystal waters instead of 1.
But the packing was different, probably. So it was a different, yeah.
This dihydrate was very stable. It was not hygroscopic. In contrast to, for instance, the monohydrate, which was hygroscopic. That was a very nice pattern.
Maybe a question back to the application field. We obviously in Basel we are at the core of pharmaceutical industry, but we have a representative of BASF. So I wanted to come back to your comment regarding battery technology, material development. Can you elaborate a little bit more where you see the advantages of this technology for the battery material development and accelerating this? And where would this play? Because of course Europe might not be one of the core places for this. Would you see this on a global basis? Thank you.
Well, I have to disagree about the European bit. I think Europe is great for making batteries, yeah. And for researching the battery production. I think there are lots of places along the line of a battery production. So you start off with a precipitation where you have really, really fine crystalline growth, yeah, of these hydroxides, and this precipitation process is extremely vital for the quality of the final battery material, right? So and there are a lot of very fancy little tricks and tweaks on how to produce the best precursor cathode active material. I'm always talking about cathode active material, right? That's the most valuable part of the battery from a chemical point of view. So this precursor material that has to be of a great quality, and the way the crystallites grow depend very, very strongly upon the mechanics and the entire chemistry of the precipitation process. And that can be studied very nicely using, for example, 3D electron diffraction. Along the line, what you do once you've got this precipitate is that you mix it with lithium, and then you heat it up and intercalate the lithium into these hydroxide layers, driving out the hydrogen, and then you get your final cathode active material. And that is an extremely crystallographically relatively interesting, very anisotropic material, right? So you've got these layers, and the lithium goes in and out of the layers. And the way that works, because you've got these little balls, yeah. You might have seen some nice SEM pictures. You've got these little balls of cathode active materials. They're made up of thousands of little crystallites, and these crystallites have a very anisotropic behavior electrochemically, yeah, because the lithium goes along these layers, and the way they are oriented in 3 dimensions makes a big difference on how the electrochemical properties of these cathode active materials are, and all along the line you can optimize and change the crystallography of these cathode active materials, changing their electrochemical properties and optimizing your material, and seeing that we do want to go away from cobalt, yeah, because of all the African children in the mines. We do probably want to get away from nickel because, yeah, Russia, yeah, and we want to increase the manganese because that's all over the place. Yeah, you can get it cheap and nicely, and but we don't want to give up the fantastic properties that we have today. So we have to engineer these materials really, really carefully, and gaining a crystallographic atomic insight into how these things are built up is quite vital.
I appreciate very much. For the sake also of the audience, which some are very on the industrial side, and I have another question for you later on the industrial side, but then also on the life science side. And actually at that stage, Christoph Boss showed us in his circle so nicely the phases of our pharma is going through in the development from early research up to the final, let me say, applications to the authorities. So at what stage do you need the structural information? Some may be very early in the, let me say, in the preclinical phase. Is there also some structural information you will still need in the clinical phase? It's more or less a question to the audience. I see some nodding here. If somebody in the audience feels encouraged to answer this, would be great.
Of course, in that case, depend on a case. In many cases, we might have challenging APIs that needs to be proved that the structure is the same all the time. And it is not often, but I met this kind of APIs that don't want to be crystalline and looks like semi-amorphous by powder diffraction. But then when you look in electron diffraction, you see that it has some peculiarities and some special structure, which is quite stable, and you don't have to be worried. And so you can show this to authorities that we don't have any worries, we don't have unstable amorphous, we are fine. This is just one example, but I think that also structure needs to be known in a formulations. So what is our, I was thinking all the time when you're talking about this patents, I was thinking about dry inhalers and what we are doing with the API, we are micronizing it. So we also introduce amorphous on the surfaces or somewhere else. And it is every day I had a question, where is this amorphous? Is it on the surface? Is it? So I think that I would like to do the electron diffraction all the time.
We appreciate that. And all the time is also, I love also the word of all the time, because actually it comes to the two questions I have to our panel. And first, indeed, we heard already about drug formulation, you know, and the later stages. How important is it in drug formulation also to use, to go back all the time again to the structure?
Yeah, well, as Anna also mentioned, so in formulation is important because if you know the crystal structure of your form that you think is the most stable and most beautiful, you still need to make sure that it doesn't interact with excipients and everything what you want to put in there. And there is also important to have a predictive morphology to make sure what groups are pointing out into the faces of your crystal to make sure that you have a good match with excipient. So it would reduce, by enabling more modeling, it will reduce the amounts of experiments that are currently done in the formulation process.
So, but this comes to the next question I had already in mind. I need to be honest, I'm trying to make a red line through the whole thing, because this, as you said, all the time, you know, this was the key word, what means how ready, or how do you see the readiness of electron diffraction for monitoring? You know, doing it all the time. You know, I'm coming now to you, or let me say, like the gentleman with his battery research, and you explain to us you're watching the crystals growing. So I assume you will take samples all the time. Maybe I'm wrong, yeah. So you have a large cohort of samples. Is it easy to work with the instrument with a large cohort of samples?
Well,
Okay. He's the right guy to answer the question. Sorry.
So to do a high throughput, of course, you need automation. Software, particle recognition. Once you can do that in the machine, then something like this is thinkable. So you're basically having a powder from your production or formulation or from your tablet, you're grinding it or put it in small, whatever, and then you leave running overnight your machine, and then at the next day you come and you know it's still as it should be. I still have 99.9% of my polymorph and 0.01% of an impurity, a polymorph or whatever. So it depends on automation, which is of course something that we will do in the future with our device.
Okay, you want to add something?
I think we might take this even one step further. Eric mentioned, you know, the vacuum or the sensitive materials. Now, if you think about your setup and the MEMS chip, you might even do what Bernd is looking for, actually do in situ diffraction. I mean, we have in situ TEM now already, but you might go to in situ diffraction. I think that would really then show you on a kind of atomic level what's happening.
Like a little bit, when we, Erik and myself, we will have a session about very practical issues at the very end and what the machine can do and so on. So I would like to draw the last 2 to 3 questions more into the very future, as you started already. And so the first question is, where do you think that the method will be in 5 and 10 years? Maybe even you give all of you a small statement on that. You start, eh? Michael, you start. Eh?
How I see it. I mean, for small molecule research, for structure determination, polymorph characterization, the key development might be the robustness of the method and to do it really routinely as we have all the time. This keyword, that is the key development. For biostructure research, I see some more severe, let's say, technical challenges with respect to data quality and really completeness of the data in order to really solve structures. Consequently, this might take a little bit more time to come. But you never know. Also with other methods, you suddenly have a jump into the development and then this turns out to be reality in a short period of time.
With Paulina, I just want to, before you start with your point, I have another question then, but after this round, which is even go further into the future, and it is about the modeling and the AI and the AlphaFold. So you don't need to focus right now on this in your statement, if I may say so, where you see still the electron diffraction, the measurement, and so in the 5 and 10 years range.
So I remember it was either electron crystallography school or one of the conferences the same year. There was this discussion what would happen in 5 years. And my conclusion was that within the 5 years, we will be having electron diffractometers. And it is happening. And next 5 years, I think that every second, maybe not every second, but more and more structural facilities like X-ray crystallography facilities will have next to the X-ray diffractometer, electron diffractometer as a complementary tool. So that is the near future, I think. And then the further I will say.
It's the modeling. Okay, no, I'm coming to that.
Yes, I completely agree. I see many electron diffractometers being spread around in the next few years, and more people getting access to it. Perhaps not in their own lab, but in perhaps sort of centralized facilities like this one, where you can easily come and get access to it. And I do think it's going to have a quite a fundamental impression on all the people that do use it, because of this fantastic properties.
Practitioners get their hands on and make their experiences, and then we will learn a lot from them, and we will learn from their mistakes, maybe, and then we can develop further, and ELDICO can come up with ED2. Okay.
Yeah, I don't know what I can add. I think it will be a huge learning curve, and people would immediately, if there is something, especially if there are problems with their batches, or immediately they will go into the detail of electron diffraction to see what is at the structural level going on. Amorphous on the surface, is the crystal good enough, not? So I think it will be a huge learning curve, and I hope there will be diffractometers everywhere. Mm-hmm. Yeah.
Okay, perfect. Just if you may.
Yes, of course. I totally agree with what all my previous speakers said, but I do see that there is probably a point where theory has to also contribute, because we now have equipment, instrumentation, but we're still facing dynamic effects in the diffraction patterns. And if we really want to get to the same level where X-rays are, then the theory has to do something. And I think at the moment we're limited to a certain molecular size to calculate dynamic effects. But as IT progresses further, I see that this will also be overcome, even if you don't calculate Bloch waves at the.
Perfect. Michael, you learned already today that there's a lot of new work coming to your law firm, if I may say so.
I see 3 possible future fields. First of all, pharmaceuticals. If the active ingredient gets smaller and smaller, from micronization to nano, whatever the term will be, we don't know yet. This will be an important field for electron diffraction. A second field is what I have seen in my own studies, when you have coated surfaces, like glasses or so. And I have seen a case where there was a patent about a coated window, and the coating was supposed to be photocatalytically active so as to destroy the dirt that would be deposed on the window. So it would be self-cleaning. And in the patent, they had difficulties in establishing the crystalline nature of the coating when using X-ray. And they only mentioned there is the possibility of using electron diffraction, but they never did. And that was one of the reasons why they eventually lost the patent. They did not characterize that layer properly by electron diffraction. So okay, so that's. And the third field was what the lady just mentioned about inhalers. There is the special category of dry powder inhalers. And there you have a so-called ordered mixture. You have cores of non-inhalable size, and then you have the active ingredient as very small particles of so-called inhalable size, and that's 1 to 5 micrometers. This might become smaller, the so-called inhalable size. You have to indicate in the patent how you define the size. And that is currently done, for instance, by mass median aerodynamic diameter. And here, electron diffraction could also be a tool in characterizing those active ingredient particles on the non-inhalable cores. That's the third field that I see.
Perfect. Okay, I promised already that we have a last question, a last big topic, even again, further outlook into the future. I mean, I could provocative ask you, are we going all to go unemployed because AlphaFold showed us that all this experimental work is done for nothing? We're doing only the modeling and any longer? Okay, but so far, probably nobody believes here in this. Nevertheless, I think, can we shed a little bit more light on this dualism between doing the modeling on the one side and doing on the other side the experiment on the other side. Where then again, okay, the CSD is a great database. They showed us already how they can use then already the work of decades of crystallographers, which put all their hard work into the CSD, and they're using this. They're using this with a lot of AI, also in that respect. Do you have a kind of a perspective on this part? Should I start with Paulina? Because you are the very expert in the model.
Well, I'm doing the modeling, but I still consider myself more experimentalist than theoretician. Because I strongly believe that the final answer we will always get from experiment or the proof. Well, AlphaFold is just a step to make it easier to go deeply into some other steps of your analysis. So with AlphaFold you have a starting model, but then the next question would be, what is the dynamic of this of the protein you model with AlphaFold? Even with crystals, you are not only getting like a static structure. You also have an access to dynamic properties of it, and this is completely neglected at the moment. This can also be a part of our analysis. And what is the most interesting is not the entire structure of a protein, but interactions with small molecules. With AlphaFold, we are not there yet, but who knows?
Yeah. And I think that also Christoph showed us already, you know, when he with his lock and his key. And indeed, you stressed very much that the ligand as well as the lock, the protein around, is changing at the very moment the ligand goes into the receptor. Yeah. So I think that's you touched a very important point. Yeah.
And this
so,
interaction and the conformational change of the body caused by this interaction cannot be predicted by AlphaFold.
And the success of AlphaFold relies on decades of hard work of X-ray crystallographers and electron microscopists to really solve so many structures, deposit the high quality structures with into the database, and then this huge database can be used with artificial intelligence, let's say computational methods, to come up with the prediction of new folds. And this is, in my view, the key understanding of artificial intelligence. It's only working with big data, so it relies on a large set of high quality, well curated experimental data. If we want to get rid of an experimental data, then we need to understand chemistry and physics and the interaction thereof. And this I don't see at the moment.
Should we continue with Christian? Because you mentioned already chemistry and physics and understanding that. That is your role, to my understanding.
I mean, many of you are aware that the Cambridge Structural Database is doing this crystal structure prediction round robin every few years. And the seventh one is just on its way. And talking about artificial intelligence, I think it will be very interesting to see whether in this round or maybe in the next round, somebody will come up with a completely new approach. I mean, now we have energy minimization. There's some people who already use the database information as kind of in an let's say analytical way, but I'm really looking forward, like an AlphaFold, that somebody will come up with now an idea to say, well, we can predict the crystal structures. And I mean, this will not take the business out of our hands, because at the end we will have to prove it. I don't think in a pattern we can say we have calculated this is very likely to appear. I think we will have to prove that it is really existing. We have to show a way of making it. So in that respect, but I see that this is going in this way. And already now they are in the field of chemistry. Of course, some of my colleagues in theory who think that they can basically calculate everything. We don't need a flask anymore. Yeah, we can just calculate, and now and then we prove them wrong. So
I appreciate it.
If you can calculate in advance by a generally known technique, then you will probably not be inventive. That's the problem.
I see. Good. Very
the invention, the real invention is an invention that you cannot calculate in advance. It must be surprising. If you can predict by computer, you have no invention.
Okay.
Normal.
Perfect. We appreciate so much the notion. We appreciate it. Michaela, do you have a perspective on this dualism modeling versus the experiment?
Yeah, well, I see a positive future. I think we will become smarter by knowing. Yeah, I think we will design smarter. We will become faster, smarter. Let's say. Yeah.
Bernd, okay, yeah. You will be the last one on this part to give you a perspective on the modeling and the. Well, I
can only agree. I think we can only get much smarter, much faster. Yeah, and it's always it always goes hand in hand. Yeah, I mean, there's no theory without the experiment, no experiment without the theory. So I think they go well together, and I don't think that there really are such antagonists, even though the protagonists of the various fields like to play it out like that. They really rely on each other extremely heavily. Yeah, and that's good. And they grow together. So I'm really happy the way things are going.
I appreciate so much, and I appreciate all the great contributions from my co-panelists over here. I thank you very much. We anticipated and also we got a few, let me say, very technical and practical questions to the device and how the device will develop into the future. We planned that Eric and myself will actually take up these questions in our upcoming next 10 minutes sessions. Before that, I thank you so much. I assume that already many in the online chat are curious to learn now, because they put in a lot of great questions around what protein, what size of the molecule, and these kind of things. So let me start, Eric. Why electron diffraction now? I mean, what needed to come together that we are now in a position really to go into an endeavor with this new device?
Yeah, we have all these technological developments that are going on. We get great detectors in the meantime, which are optimized for electron diffraction. We make great use of that. We managed to find suppliers that give us this great, robust, very accurate goniometer that enables to keep the sample in the electron beam all the time, which was really a great effort to find the components, to put them together, to tweak the goniometer, to keep the sample really into a sphere of confusion of a few hundred nanometers. So that was more than a factor of 10 better than what was possible on an X-ray diffractometer. And also with the lenses and the optics. Of course, the source itself, the LAB6, that's standard. But having the lenses and all the technology of the lenses and our great designer that gave us a huge benefit on the concept of the electrical optical lenses was a big step forward, mainly in making the system easier to use, simpler to maintain, more stable over time. So these were great advantages that were only possible through this technological developments.
How important was it to leave the classical TEM and really to make a new dedicated design for the electron diffraction?
Yeah, that was in some sense easy for us because we didn't come from the electron microscope side, and I appreciate, of course, the feelings that electron microscopists have, knowing their instrument, having worked with electron microscopes for many years, having developed the method and found now a proper way in getting the structures done. We will not make a big point out of it in re-educating electron microscopists. There's no sense in doing that because it's a established technology. It gives the results as they're coming out. We three of us were chemists, crystallographers, so we came from the crystallography side, and we saw the potential of electron diffraction, and I think our most trivial, but in the end also most potential thinking was that why not exchange the X-ray source by an electron source? And in the end, that's almost what we did. Of course, it had a few things to think of. The sphere of confusion needed to be changed. We needed a different detector. We needed vacuum, which was a mess, but that's the situation. So that's how we came.
Okay. Also, let me say, coming from a little in line with what the online audience wants to know, how sensitive the crystals or the compound can be. Yeah. Also in terms of what you then do to stabilize it.
Right, yeah, there are sensitive and less sensitive samples. We all have experience in that sense. We talked about sulfates and hydrates, which have their particularities. In general, organic samples are much more sensitive than the inorganic, mineralogical, geological samples, or whatever you have, concrete. Yeah, the biological organic samples are the most sensitive, and we, although we didn't plan to go for the organics right away on developing our instruments, we realized how important the organic molecules are. So yeah, we took already in developing the instruments great care in increasing lifetime in the microscope.
Perfect.
And that was done by, for example, taking STEM images instead of conventional TEM images. So we scan the beam over the grid within a second. So any crystal is hit by the beam only like a millisecond or something. And therefore the irradiation, the damage, the exposure to electrons is very much reduced almost by a factor of 1,000 already by going from TEM to STEM. We don't do continuous video imaging kind of your sample, which, you can consider that as a slight drawback. We take a still image, which you then analyze. You pick your crystal, and then you move to that crystal. You take at higher magnification another still image. But that means that before you start your experiment, your crystal have been exposed only to 5 or maximum 10 still images, and a crystal would only have gotten 10 milliseconds of exposure of the beam. So your crystals are virtually fresh when you start the experiment.
And then how long does the experiment take? How many milliseconds the crystal has to be stamped?
Although we run really at low dose the experiments in small microamp settings to reduce the dose on the sample during the experiments, we still done typically in a minute for a complete data set, 140 degrees, which is very often sufficient. It's not always 100% complete data, obviously, but very often it's sufficient to get your structure. We have done many structures from a single run, single crystal. Obviously, as a decent crystallographer, you go for completeness, 100% completeness, and then you move to the next crystal. That's another minute, and another crystal, another minute. And then you merge these data together and you get
okay, so for the audience, you need to tell this is still on room temperature. What is about lower temperatures? Kind of cryo-EM?
Cryo we will do, of course. That was clear from the beginning. We have the concepts done. We are starting to realize the development, the setup, and the creation of a cryo device. We have very interesting ideas for that. That looks very promising. In the meantime, we have been running without cryo because we didn't have any, and our experience was great in running samples even at room temperature. Yeah.
Silly question, just for my very personal curiosity. Can you also manipulate the environment? Let me say the air, the gas, giving some water molecules into the let me say a high humidity whatsoever.
Yeah, that would be this idea with this sandwich grid, the double grid. That would be one way to approach that, and we're almost there to do the experiment in the next 1 or 2 weeks. Another way is flow cells. A bit similar to the X-FEL setup, where you would have either many crystals or at least work in an enclosed cell, either with liquid or in a flow cell that has been demonstrated to be possible on the electron
put some
microscopes.
Special compounds into this liquid.
Yeah.
Perfect.
Yeah.
I appreciate that. There's a large open space to play, let's say that way.
That's one of the advantages of our setup. In contrary to an electron microscope where the sample is tied between the two condenser lenses, you have these huge lenses which are only a centimeter apart, I think. And in between you have the pole with your sample, so there's almost no space to play around with your sample in that area. We have several centimeters from the crystal to the pole shoe of our last lens. We don't have any lenses on the other side, so we have this huge space to have not only have a very rigid goniometer that holds the sample, but also to approach any additional setups. The sample chamber that we have the goniometer and the sample in is an octagon, a very accessible setup with windows, so to say, flanges where you can approach the experiment, for example, by a laser beam to photostimulate your crystal in the electron beam. So all these setups, monitoring or activation of your sample can be done through the sample chamber.
Perfect. And how big? You know, that was a question also from the audience, but also I think it's an obvious question. How big can the molecules become so that you can still measure them? For example, Paulina, she talked not only about peptides of a few amino acids.
Yeah,
it was growing and growing more into proteins.
Yeah, the molecule can be as big as it likes, as long as the unit cell. But no, that's goes a little bit together. Yeah, you need at least one molecule in one unit cell in triclinics at 1Z1. But the unit cell, yeah, I would say in ideal cases you can go up to 100 angstrom. But that's that's for the moment it's calculated. We have run one sample at 70 angstroms. So yeah, we're quite happy with that number. That's a decent organic molecule that finds place in 70 angstroms. And yeah, there's no reason to expand that. And of course that would be sought for a second model instrument that is all in our minds already, and that's why we also need more money to. So if there are any investors, yeah, further steps to develop the instrument. Okay. Yeah, it's not a huge thing to go to the biomolecules. It would require in that what we think as the simplest solution. An additional lens on the secondary side where you can widen up your diffraction pattern. Once the reflections come too close for the large proteins, we just spread them out again. You would lose on resolution, obviously, but that's typical for protein samples anyway. They don't diffract up to 0.8 angstroms as we can measure right now. So you probably throw out empty space anyway with your lens, and in that sense, I think we have a very feasible solution for the Model 2, the ED2.
Yeah. I see. Okay, and then for developing the device, your colleague also was mentioning already when we were asking about monitoring. What is the plans to for automation?
Oh, that I can't tell.
No,
there are ideas for automation. Yeah, starting from the fact that we can easily mount several samples at one time in the device. So instead of inserting only one grid, we can easily insert 3 or 5. I don't know. We need to think about solutions. But we can, with the goniometer, we can move over larger translations, and we have the space also to adapt for that. That would be one thing. You can think of changing your grids inside the machine. The most trivial solution would be a robot or a sample changer in front of the instrument for automation. Yeah. That would be the mechanical things, and yeah. In software, we just have an opening out for 2 software engineers, which we need urgently to exactly work on these things, automation of as Gustavo mentioned, we need particle recognition. What is a crystal? What is not a crystal? Or particle? In the end, it might be an amorphous particle. That's up to us or the machine to decide. But we need a device that picks up the interesting pieces against the background, and then drive the goniometer and make an experiment and process the data also on a much more automatic way. So the whole pipeline for data processing needs to be automated as well. Data processing. Yeah.
Last question, also related to that, more work for your data engineers and so on.
Yeah.
In a way, how important is the computational part and the development? Because then you get your pattern, but you don't have a structure yet.
Yeah.
Now you want to also to calculate a structure from that.
Yep. Yeah. There we are in the lucky position that we can almost apply one-to-one the X-ray software which is around, and that's why we didn't even put an effort in. Crystallographers, at least, they all have their preferred packages already in-house from the X-ray facilities. The raw data that comes out is 100% compatible. We provide many different frame formats that can be converted or stored or read into the X-ray packages immediately. We can do some smoothing up in the interfaces, the data transfer. That is still a little bit homework for us, but we rely 100% on the.
Okay, so that work is done by the X-ray guys already. It has been done already by the X-ray guys.
Up to
Michael, great. Okay, so. I would say we close here because, honestly speaking, we are already half an hour over time.
Getting thirsty.
And I don't know if the liquid nitrogen above on the molecular cuisine is still fresh,
still cooking,
and still cooking something. So he's definitely waiting on us. I appreciate very much all your patience and your attention to the panel and to the talks.
Yeah. Exactly.
And
you're very much invited. Very much invited to the upper row, as we say in Switzerland, with a little bit to drink, a little bit to bite, and I thank you very much for your attendance, keeping through all the afternoon and investing your time. I hope you liked it. During the upper row or even here, you can subscribe for a short demo, 15-20 minutes demo on the machine downstairs on the ground floor. We will go down in small groups of 5 people, I would say, and that will take 15-20 minutes. So whoever is keen on seeing the instrument in operation, we will show a little bit of measurement and things like that. Is happy to sign up for the demo downstairs. But otherwise, let's meet at the upper row, which is at the 5th floor. Thank you.